Welcome back to my channel Xgene and proteinX. In this tutorial video, I'm going to talk about DNA replication in prokaryotes, and in this session mainly I'll focus on initiation. So let's begin. So these are essential for DNA replication in prokaryotes. These are DnaA, DnaB, DnaC, DNA polymerase 3, and DnaG, also known as primase. So let's have a look at the structure of the DnaB, DnaC, and DnaA. So DNA, it contains 4 different domains. DNA 1, 2, 3, and 4. DNA 3 also it divided 3A and 3B. So DNA 1 and DNA 3 it interact with the DnaB, and DNA 4 its main function is DNA binding. And next is DnaB. It also contains the 4 domain, and these numbers are representing the amino acid residues. And these domains it interact with the primase, and this domain RECA domain it interact with the ATP and DnaC. So next is the DnaC. It contains 2 different domains and it interact with the DnaB. So next I wanna show you the origin of the replication. So this is the circular DNA. As we know, the bacteria contain the circular DNA. And here I'm going to show you the initiation process in the E. coli. So in all of the bacteria, their initiation steps or the DNA replication process are all the same, but little bit varies in different species. So in the E. coli, in the origin of the replication or ORIC, it contains 2 different parts, DUE and DAR. So DUE, it is a DNA unwinding element. So it contains 13 mark. So 313 mark, and each contains 13 amino acid residues. Those are this sequence. This is this sequence, and also it contains the AT rich region. So this percentage of the adenine and thymine is higher in this part. So this is why the hydrogen bond breakage of the hydrogen bond it's very easier. So unwinding occurs in this place, in this region. So another part is the DAR, that is the DNA assembled region, and it contains 5 different boxes. So these boxes are actually, this is known as the 9 mer. So each contains 9 amino acid residues. So this amino acid residues, this one, and also these boxes are known as the DNA box, because this DNA box you see here. So because the DNA bind with this DNA box, this sequence. So this is also known as the initiation recognition sequence, right? And also another thing is that it contain the E. coli ORIC contain IHF and FIS, this binding site. So let's have a start the initiation process. So DNA first is the DnaA. It recognizes the specific DNA element in E. coli ORIC, that is the initiation recognition site. So it recognizes the initiation recognition site where the initiation start. So and then this DnaA it forms the oligomer assemble. So this is the assemble, and this Roman number is representing the functional domains. So then it forms the DNA ATP assemble, and it induces the unwinding of a region containing 13 mer. So this DNA ATP it bind in the upper strand of the DNA here, but it's not showing here, and then it induces the unwinding in this region. So at this step, the domain 4 of the DnaA it interact with the DNA boxes. So the DNA 4 contain this domain, mainly this domain, it interact with these DNA boxes. So next is the domain 1. So and then this unwinding it start, and then the replication fork it formed. So you see, this is it looks like the replication fork. So next is the domain 1 of the DNA that interact with the DnaB, and that DnaB it forms a complex with the DnaC, and then this DnaB and C complex it loaded in the each unwind DNA sequence. So this DnaB-C complex it loaded the unwind region of this DNA sequence, and then it the negative supercoiling it occurs, and the unwinding it starts by this DnaB and DC complex, because they are putting pressure in opposite direction. So next is the primase, that is also known as the DnaG. It interacts with the N terminal of the DnaB. Alright, and this primase is required for the primer synthesis. So next step is as primase synthesis, and a primer is actually when the primase is interact, and then primer synthesize in both of the strand, upper strand and the lower strand. So then the dissociation of the DnaC occurs. So whenever the primer it synthesizes, then the DnaC it dissociate from the DnaB. Alright, you see the DnaB is only alone here because DnaC already dissociated. So next step is the primer synthesis part. So primer already synthesized by the primase enzyme. So next is the primer that synthesizes, and that primer it transferred to the DNA polymerase 3, and upon transferring this primer to the DNA polymerase 3, then the primase enzyme is dissociated from the DnaB. And the next step is the DnaB. This moves on each single stranded DNA toward the junction of the application fork. So next step is the DNA polymerase 3. DNA polymerase 3 it extends these primers for the synthesis of each leading strand, and then the single-stranded binding protein, those act here in this region to prevent the secondary structure formation, and also prevent the degradation of the DNA strand. So next is the primer it synthesizes for the replication of the Okazaki fragment. So that is the elongation part. So I will talk later, and I will make another video for the elongation. So this is all about the initiation part of the DNA replication in prokaryotes. So I hope you like this video.